Hello, I'm Maruk Said, your microbiology guide. Let's explore in-depth micro techniques and uncover groundbreaking insight together on this educational journey into the fascinating realms of microbes.
Have you ever thought about how ancient viral DNA remnants from the viruses that infected our ancestors millions of years ago could still be active today? Even more surprising, these DNA fragments, known as transposable elements, play a crucial role in the very beginning of life, which is early embryo development. Hi, I'm Maruk Said Gilani, and today we are diving into the fascinating world of ancient viral DNA and how it shapes life as we know. Let's unravel this together. First, let's understand what we are talking about. Did you know that over half of our genome is made up of ancient viral DNA? These fragments, known as transposable elements, were once dismissed as junk DNA or the dark side of the genome. But new research from Helmholtz Munich and Ludwig Maximilian University has flipped the narrative. These elements are anything but useless. They are a crucial player in the earliest moments of life.
If you have watched till here, don't forget to subscribe to Family of Science Lovers. Press the bell icon button so you are always updated.
After fertilization, the embryonic cells begin to divide rapidly, and during this period, transposable elements are reactivated. They become part of a complex network that governs cell plasticity, the ability of these cells to become any type of cell in the body. But this raises some big questions. Why are these ancient viral elements reactivated? Are they universally important across all mammalian species? And what mechanisms regulate their activation? These are mysteries scientists are still trying to solve. The research team led by Professor Maria Ellen Torres Pendela developed innovative methods to dig deeper into these questions. By understanding embryos from multiple species like mice, cows, pigs, rabbits, and Rhesus macaques, they created a groundbreaking data set that's shedding light on these ancient elements. Here where it gets even more exciting. The team discovered that viral DNA elements thought to be extinct are actually re-expressed in the mammalian embryos. Each species has its own distinct set of active transposable elements, but their activation appears to be a common feature across mammals. It's like uncovering a genetic signature of early life. These findings are huge because they open up new doors for understanding cellular plasticity and even manipulating it. Imagine being able to guide stem cells differentiation by targeting these elements. This could revolutionize regenerative medicine and developmental biology. But why does this matter for you and me? Well, understanding how these elements regulate early development could lead to breakthrough in the reproductive medicines. For example, fertility treatments could benefit from insights into how embryos regulate these ancient elements. Additionally, manipulating these elements could help us understand diseases where cell plasticity goes errors, like cancer. It's like small pieces of genome with enormous potential for health and disease research. One of the most remarkable aspects of this study is its evolutionary perspective. By comparing transposable elements across species, researchers identified regulatory pathways shared among mammals. This helps us understand not only our own development, but also how these mechanisms evolved over millions of years. The study also generated a dataset that will be invaluable for researchers worldwide. It's like having a roadmap to explore the earliest stages of life across different mammals. So what's next? Professor Torres Pendela's team is now focused on understanding the specific regulatory principle behind transposable elements. By decoding how these elements are activated and controlled, we are paving the way for future breakthroughs in the science and medicine. This research not only enhances our understanding of early life but also shows us how interconnected our biology is with the history of life on Earth. Ancient viral DNA might sound like a relic of the past, but it's very much a part of our present and future. For shaping early development to holding the keys to medical breakthroughs, these transposable elements are a testament to the incredible complexity of life. If you enjoyed this deep dive into the world of ancient DNA, make sure to give this video a thumbs up and subscribe for more fascinating science content. Until next time, stay curious.
Terminologies. First, transposable elements, TEs, fragments of ancient viral DNA embedded in the genome. Once considered junk DNA, they are now known to play critical roles in gene regulation during early embryonic development. Second, cellular plasticity, the ability of embryonic cells to transform into any type of cell in the body. TEs contribute to maintaining this flexibility in the earliest stages of life. Third, fertilization, the process where a sperm cell fuses with an egg cell, forming a zygote that divides into embryonic cells. Fourth, embryo development, a rapid cellular division process, post-fertilization, during which TEs are reactivated and influence the fate of the developing cells. 5. Ancient viral DNA, genetic material from viruses that infected ancestral species millions of years ago, now integrated into the genome. 6. Evolutionary biology, the study of how species evolve over time. Comparing TEs across mammals reveals shared regulatory mechanisms and evolutionary pathways. 7. Regulatory pathways, networks of genes and molecules that control when and how TEs are activated during embryonic development. 8. Stem cell differentiation, the process by which a stem cell develops into specialized cells, like muscle or nerve cells. Insights into TEs can help manipulate this process for regenerative medicine. 9th, reproductive medicine, a field of medicine that focuses on fertility and embryo development. Understanding TE regulation could lead to improved fertility treatments. 10th, regenerative medicine, a branch of medical science focused on repairing or replacing damaged tissues or organs, potentially guided by insights into TEs and cellular plasticity. 11th, comparative genomics, a method to study similarities and differences in the genetic material of various species. This helps identify universal biological mechanisms. 12th, dataset, a collection of organized data. The study generated a comprehensive dataset of TE activity across species, a valuable resource for ongoing research. 13th, embryonic cells, cells formed after fertilization that eventually develop into all tissues and organs of the body. 14th, species-specific TE activation, the unique set of TEs reactivated in embryos of different species, revealing genetic diversity and evolution. 15th, cancer research. TE studies can inform how cell plasticity malfunctions in diseases like cancer, aiding in targeted treatment development. 16. Helmholtz Munich and Ludwig Maximilians Universität, the institutions where groundbreaking research on TEs and early embryonic development was conducted. 17th, evolutionary tree, a visual representation of relationships among species, highlighting shared evolutionary traits such as TE regulation. Thank you so much for watching. Don't forget to subscribe to my channel, press the bell icon button. Thank you!